biology, topic by topic. This time we're going to have a look at drugs. And immediately when you talk about drugs, people think, oh, cannabis, heroin, the evils of drugs. But in fact we're going to look at the other side of drugs, the useful drugs, the ones that are developed for basically treating people. So drugs come in basically, let's say, two different categories. Something that's actually going to work on and kill the bacteria, the virus, whatever we want to try and kill, and the other one just make us feel better. And this is one of the age-old problems about curing the disease or curing the ailment, I suppose. If you can make your headache go away, are you really better unless you've actually stopped the cure of the headache? Let's have a look at some ideas here about really treating the symptoms. So, we've got basically two choices. We've got some sort of drugs, and these might be from one of the earliest ones, aspirin, paracetamol. These are sort of, when I was a child, I suppose, aspirin was the most common sort of painkiller because things like paracetamol hadn't been yet fully developed. And now we've got other drugs like ibuprofen and so on. Now, let's see, what do they actually do? Well, these basically perhaps sort out a headache. They can relieve pain. So if I've got some bacteria growing and that's what they're causing, then that's the sort of thing that we're actually trying to fix. But what kills a bacteria? Let's have a look. Killing a bacteria is relatively sort of difficult. If we've got our little bacterium, what it does to grow to become more bacteria is it's got a ring of DNA and some plasmids and some other bits and pieces in there. This ring of DNA duplicates and the cell breaks down in the middle, and then it grows, and then it can do it again. So what we need to try and do is to try and stop it do that. Now, stopping the replication of the DNA could have some nasty side effects. Yeah, have you thought what they are? Yeah, it could kill you as well. So what we try to do is that because they've got a special bacterial wall which is different, what we have is an antibiotic. Now, an antibiotic, and there tends to be not many of these. The first one, oh, spelt that wrong. Penicillin is a family of drugs, and this family of drugs, what they do is they interfere with the cell wall here, and so they basically punch a hole in it. All the insides of the bacteria fall out and the bacteria dies. So what can we do? What are the problems? Well, the problems are that we haven't got very many ways of trying to kill a bacterium. Basically, we managed to discover a drug, penicillin, and now there are a family of penicillins, similar drugs, very slightly different, and if the bacteria can be resistant to one, maybe then one of the others might work. But we've only got a limited number of them, and we can't seem to develop any more. Why not? Well, taking a drug, making it, takes a lot of time, and we've got to look and see whether it actually is going to harm people. So we have to worry about that. So certain bacteria are more susceptible to certain drugs. So we have our penicillin family of maybe 3 or 4 different types of penicillin. You may have heard of some of these. Tetracycline, ampicillin, cephalosporin. We've got several of these different drugs. Now, this might be more effective against certain bacteria, and it can kill off these bacteria, whereas another drug might work better on other bacteria. They're more susceptible to them. So our first job is to have a look at how we can destroy bacteria, and one of the things that we can do is do a basic test on these. So let's have a go and see how we have a go at a test on these. So let's take a couple of agar plates, and in these agar plates we're going to cover this with what's called a lawn of bacteria growing. And what we do is we put down some antibiotics, and we put them on a little paper disc. So here, this is simply a paper disc and this soaked in some sort of antibiotic. And what we do is we allow the bacteria here to grow, and we've got this little disc. Let's see what happens. Well, there's a picture of this sort of thing, and we can see in the one on the left that the bacteria is being killed. The clear areas are where the bacteria are literally being blown up, proper word, lysed, and we can see quite a lot of damage, quite a lot of dead bacteria suffering from these antibiotics. But the one on the right, we can see that some antibiotics are working and some aren't. Some of these bacteria are resistant to these antibiotics, either naturally because it's not designed to kill them, or sometimes because they've managed to get some genes that can protect them from these antibiotics. And that's becoming a worry. Why is it a worry? Well, simply a disease that you've had, that you were cured with an antibiotic, your children or your children's children might not be, because the bacteria have become resistant, and then when you give them this antibiotic, it doesn't work, and you've got nothing to try and prevent them from dying. So an antibiotic here will give us a sort of a ring around ones that work, and then if we know that works, then we could try this antibiotic or that one, or maybe even sometimes a combination of the two. Our big problem with an antibiotic, though, is really quite simple. Is how do we get it into you? Yeah, okay, so we can take a tablet, but that tablet then goes down into the stomach, and when it gets down into the stomach, then we've got a problem when it gets down into the stomach, that we don't want it basically destroyed by your enzymes. It needs to get absorbed. And once it's absorbed, then we can go into the bloodstream or where that bacteria is. But we have another problem. So let's imagine that we've got here, let's have a couple of views of a microscope slide, and in here what we've got is a lot of bacteria growing, and the bacteria that we've got growing are these little blue ones here. These are your little bacteria, and these are the ones we want to kill. But in here we've also got some other bacteria. These are, let's say, good bacteria, and these good bacteria are bacteria that basically you find are useful in your body. They may digest something, they may do something else. But in here I've also got one bacterium that is resistant. So what happens here when we do some damage? So we take an antibiotic, and now the only one that's left is this one. Well, over a period of time, this one now replicates. It's got no competition, so this bacteria can now replicate in your body. So we've gone from having just one that was resistant, with the use of the antibiotic that didn't destroy it. This is now taking over. Why didn't it take over before? Well, it didn't take over before simply because there was so much competition. You removed the competition. This is survival of the fittest. And now you've got lots of these. And we can find that there is one other little problem that we can meet with this, and that is that this bacterium has a nasty habit. This bacterium can now find another bacteria, and it might give it the antibiotic resistance. So now if we do that same scenario, we have a slightly different problem. So we've got some of those antibiotic resistant bacteria. We've now got some other pathogen that has been given this resistance, and you've got a few good bacteria, I suppose. And when you now take the drug, we simply get no change. We have no effect. And we have, within all likelihood, death. Bit grim, isn't it? So we have to worry about bacteria. We have to worry about use of antibiotics. We find that antibiotics are not just used for basic uses of sort of rescuing people from a disease, but we can also worry about it for another way, because we may just put it into the feedstocks for animals. Now, if I give them some food plus I give them some antibiotics, then what we've now got is healthier animals, that they don't go down with that bug. But the big problem is if you don't finish a course of antibiotics, and we had that little picture up a moment ago, which I'll put up again. If we look at one of these discs, and the one I want to look at is this one here, this drug is working, but only at high concentrations. This one appears just to barely work on some of them. Now, if you take your full course of antibiotics, then the likelihood is that you're going to probably kill all of the bacteria. You're not going to leave any odd stragglers behind, because as soon as we leave the odd stragglers behind, they are going to say, okay, you killed off all the opposition, I can grow now, and I can grow, and I've got this extra antibiotic resistance. So we always try and carry on the course. If, however, we've got bacteria that are resistant, and then these now enter you, they're antibiotic resistance, and the antibiotic has no effect. The other problem with antibiotics is basically they work on bacteria really quite well. But let's suppose you've got a virus. Well, I'm sorry, they have literally no effect, because these are inside your cell, whereas the bacteria are outside. So it's very, very difficult trying to develop drugs that will kill viruses. We can. There are quite a lot of drugs that will kill viruses. The technical problem is they tend to have side effects, like they kill you, and it's not really good. We've then got other bugs, and one that comes to mind is this methicillin resistant Staphylococcus aureus, MRSA, which is tested for when you go into a hospital. Many hospitals just test for this straight away to make sure you're not trying to bring this into the hospital. If you are, they will treat you, they just have to treat you differently, which is probably a good idea. So these are a matter of great concern. You may well have MRSA, and that isn't necessarily a problem, because you have MRSA and naturally you can kill it, because it isn't just antibiotics that do the killing, but we have various other things that will kill them. So let's hit some words. We've got an antiseptic. This is something that will kill the bacteria, microorganisms in the environment. You wipe down the surfaces, you kill them. We've got an antibiotic. This will kill bacteria in the body. And we get antibodies, and these are made by you by the white blood cells here, and they will destroy both viruses and bacteria. So this is the route to go if we can. This is perhaps through the routes of things like vaccination to try and make people a bit better. But we can't always rely on having a suitable drug. Our problem is that if we've got a virus, then this is typically inside the cell, and even antibodies can't really attack at that point. They can only attack normally outside the cell. So we've got two things here that we've been talking about. We've got the idea of a painkiller. This destroys the pain but doesn't kill the bacteria. And we've got an antibiotic that does kill the bacteria, but more and more we're finding these becoming less effective. So what are we going to try and do about this? Well, what we're going to have to do, I suppose, is discover some more drugs. And discover is probably the right word here. Now, where do most drugs come from? And the answer is they come from plants. Plants are being infected by bacteria, funguses, viruses, and the plants over the years produce different defense mechanisms. Many of the drugs we use today are based on traditional plants. The story goes that you have, in old times, men going out doing all the work and the women staying behind doing the washing, the cooking, the cleaning. And no drugs, no medicine. And you've got the women down by the river doing the washing. Yeah, I know it's very sexist. And a young girl started a period and having pain, and one of the mothers would say, yeah, go down there, get some willow tree, pull off a bit of bark and chew the bit of bark. So they do, and the pain goes away. And that is because inside that plant it's got a chemical, a drug, salicylic acid, and that is designed to try and prevent this plant from being attacked by microorganisms. And it also has the effect, it's a very good painkiller. It's aspirin, basically. Now, that's an anecdotal story, but based on the truth that we've also got various old wives' tales, as they're called, where people have got this folklore going down through the generations that if you do this, if you try that, then this will solve this problem. Some of it was wild and sort of a load of rubbish, but a lot of it was actually based on some sort of truth. And a lot of that was based on these women, these witches, who would manage to make concoctions from different plants to alleviate some problems. And many of them really did work. Those women, of course, that survived the witch hunters and all the other bits and pieces that were going on. So what we need is these drugs, and we find things like the willow tree, and from that we've developed the drug aspirin, and that's still used really today, largely for controlling heart disease. Round about the early 20th century, people started working on trying to see if they could actually isolate some of these chemicals and work out what they could do. And in 1928, a guy called Alexander Fleming, famous that he was looking at bacteria, he was careless, he wasn't a good scientist really, and he found that one of his cultures had a mold growing on them, and this mold was the penicillin mold. Penicillin mold's actually used in blue cheeses. Not necessarily the same variety, however. And they tried to extract this penicillin about 10 years after Fleming's discovery, and this is where he had this mold growing and the bacteria not growing. A couple of gentlemen, Ernst Chain and Howard Florley, set out about trying to extract the penicillin, and they managed to succeed, and they managed to sort of give somebody with a big blood infection some of this penicillin mold, and it worked. Unfortunately, it didn't make very much, so it worked for a while, and then the guy got better, and then he died because they didn't finish the course of the system. But they had found out that they could make penicillin, and drug companies worked on this. And once they got these drugs, they basically could work on things to sort of make the people better. Now, some bugs are resistant to this, so we need to find new drugs to try and do something. So we can look at here different varieties of penicillin, that if something will stop this working, if you've got a slightly different variety, then maybe that won't be stopped. And to a certain extent, this has worked. There are a whole series of different chemicals like this, and we've been working hard on trying to extract them and try them out. But there are problems with these things. Now, all the time we're discovering new diseases, and as we're discovering new diseases, then we've got to find something that's going to try and be fixed. So many drugs are based on plants, and then what we do is we sort of try and change them. So this is what the pharmaceutical industry does. It takes perhaps an existing drug, modifies it, and we have ourselves a new drug that can then be developed and tried out. We've got quite a lot of drugs that we can try out, and there are all sorts of things. You've probably heard with COVID-19 they're trying different existing drugs. Now, the reason for using an existing drug is that you know it's been tested, you know it's been safe. It's safe to take, because some of the side effects on some of these can be rather lethal. And so we've got to try and go through a mechanism of finding a drug. So finding a drug, well, it's got to have certain properties when we look for a drug. Firstly, it's got to be effective. In other words, it must actually prevent the disease or cure the disease. It's got to be safe to take. In other words, it can't be toxic and kill you. Or, I suppose, let's have side effects. It's got to be stable. Basically, we've got to be able to store the medicine, and basically you've got to be able to use it under normal conditions. So it's got to be here, and basically one other thing it needs to be taken in and removed from the body, because it's got to be able to be removed, because if it can't be, then we're just going to build up resistance to this particular drug. So how do we go about developing a drug? Well, it's really very, very simple in theory. Unfortunately, it takes a lot of time, and this is the problem in practice. So let's suppose we want to find a drug. So let's start off with a disease, and having got a disease, we then basically try and find some drugs that might work on this, and we're going to test a whole load, let's say 5 to 10,000 different drugs we're going to try on this, and some of them are going to work and some of them aren't. We try all sorts of drugs on this, and we start with all the ones that are known plus a few others that don't do anything at the moment that we know about, because they might be the drug that's going to work on what we're actually trying to sort of destroy. So we start these drugs and basically we test them on against these viruses, bacteria, whatever, and this process takes a long time. So 4 to 5 years might be taken to try all of these drugs on each of these different diseases that we're trying to sort of have a go at. And many of these we've got to try and do in the lab. Why we're trying to do this in a lab is because we can't afford to do this on people, because many of these drugs might actually kill the people. So this is the pharmaceutical industry working hard. 10,000 drugs working on this particular disease, this particular bacterium or virus or whatever it's going to be. And when we're playing around against COVID-19, people want this brought down to months or days. Well, we do have the ability to cut this list down to a certain extent. So we've now got to a stage where we've got some possibilities of some drugs. Let's say we've got now between 10 and 20 drugs that show some promise. Right, so what can we do against these? Well, we can try what we call preclinical trials. So here we are now testing these on cells. We're selling these on tissues, all grown in the laboratory, and maybe some live animals. And this process is not quick. This takes 1 to 2 years to actually do. And coming out of this, we've now got perhaps 5 to 10 chemicals that show some promise. So we now go on to clinical trials. So things here that haven't killed anything, then we now do a clinical trial. And what we do is we take a healthy person, someone who hasn't got the disease, and we give them a very low dose. And what we're looking for is to see what problems this may have. Now, this is not without its risk. These people are paid well for doing this, and that is because the side effects could be disastrous. They could even kill. Just because it didn't kill an animal doesn't mean to say it's not going to kill a human. So we try this on low doses, and gradually they try a few higher doses. So we've now tried it on healthy people. We've given them a low dose. It seems to be okay. And this process does take, again, some time. So maybe 1 to 2 years to do this. And we're down now perhaps to 2 to 3 sort of chemicals we want to try out. And we test those. And we do some more tests on these. And what we want to now do is to try and find out how well these might work. And so our next idea that we can do are things like blind trials. So what's a blind trial? Well, this is where the doctor knows whether they're giving you the drug that we're testing or perhaps an existing one. And the patient doesn't know. And that can give some ideas about how effective the drug is. You can compare it with the existing drug and you can see whether it seems to be working. You can then move on to double-blind trials. And in the double-blind trial, neither the doctor nor the patient knows whether they've got the drug, or it could be a placebo, it could be no drug at all, but that's not really ethical. So what often is used is an existing treatment, and they can compare an existing treatment with a new treatment. And sometimes the doctor doesn't need to know, but sometimes they do. So sometimes we can do double-blind trials and sometimes we can't. And we basically come out with one drug that seems to be fairly effective, and this then needs to be licensed. And once it's licensed, now this is safe to be going out and used on generally the public at large, I suppose. But look how long it's taken. Maybe 5 years, 7 years, 9 years, 11 years. And for the licensing, we might be talking here up to 15 years. If we're looking at COVID-19, we're looking at some of this being accelerated to try and get into some of these clinical trials after not this number of years, but after 3-4 months. They're trying existing chemicals because they know those are safe. Then they can try some other ones. And if you're in a position where you've got this bug, you're going to die, the doctors say, yeah, look, we've tried everything we know, we can't do anything for you, but there is this new experimental drug. No, we haven't tried it, we don't know how dangerous it is, it might kill you, but you're going to die anyway. So people take the risk, and this way we can move things forward. Yeah, it may work, and there's always good signs that I've got a drug, yes, it's going to work, but it doesn't have to. Luck doesn't work that way for many people, and we have to sort of worry about, whether a drug will be effective or whether it won't be. And people have to sort of put their lives on the line. They have to say, right, I'm going to try this drug out, and it may or it may not work. Big problems here. Once we've got a drug, it's licensed, everything about it is published. So not only one person knows about it, the drug company that developed it, but now it goes out. And this is where there's a little bit of a problem, because if you're a drug company and you've spent millions, billions perhaps, on developing this drug, and then you just have to give it away, you don't recoup all that money you've spent out. And drug companies, like any other company, are trying to make a profit. They are, yeah, profiting on people's lives, but they're also employing lots of people. And if they can't produce the drug, then they've just wasted that money. So it's not that unfair to expect them to get their money back. But once we've got a drug that you get a particular brand for, so when sort of a particular drug came out, it sort of comes out with this brand name, and then after a period of time, then all the other drug manufacturers are allowed to produce this drug, and you see the cost come right down, and the popularity of the drug become more widespread and easier to get. So we have all this thing with drugs and the way it works, and whether you feel it's right or not is sort of really irrelevant, really, because we've all got to try and make some money. And if you don't let the drug companies make any money, which is what a lot of people think, they're making obscene amounts of money on this, then the drug companies go out of business. And if they go out of business, there are no new drugs. It costs a lot of money to get a drug. We're looking at 15 years. We're looking at quite a lot of people working on this for a long period of time to bring a drug to the market. Once the drug's been out there, lots of other people will try and test it. And this process of trying to license the drug does in fact take a while, with other people trying to test it to try and make sure it works, and it works safely. So there we are, drugs. They're not as bad as they seem. The good drugs are the ones that are going through this system of being tested. Now there are some other drugs you can buy on the street. Now these haven't been tested, and you don't know often what's in them. And maybe that if you take a drug and you mix it with something else, it can have a particular side effect. So we have to be very careful on drugs. Prescribed drugs, they are licensed, they are made to be safe. Whereas there are two classes of drugs that aren't. The first are the ones that you all know about, the sort of ones that are sold at the back of the school, the ecstasy, the other hard drugs. But there is one other group of drugs that seems to miss this, and that are the drugs about vitamins. Now vitamins are good for you, aren't they? They're not harmful. Or are they? So we do know that with lots of drugs, like these multivitamin things, that if you don't have them, you can get problems. You can have, like vitamin C, for instance, you can get scurvy. You can get vitamin B, vitamin D deficiencies. We see these diseases. So many people think, right, I'll solve this problem. You haven't been that well, you haven't been that healthy. What I'll do is I'll get you some vitamins. Now, in fact, we normally eat more than enough vitamins in our normal foods that we eat. So what happens if you take too much in the way of vitamins? Well, this is a part of the world that's not really very well licensed, and some of these can be dangerous in certain circumstances, because we don't need very much, and if you take too much, generally your body gets rid of them. Sometimes they store them up, and that can sometimes lead to being a problem. Whereas people don't tend to die of too much vitamins, there are certain problems that we meet with some of these vitamins when you have too much. So something you have to be reasonably careful about. Do you need vitamins? Do you need to take a vitamin tablet every morning? And the answer is probably, if you have a good diet, no. Is it a waste of money then? Yeah, probably. Can it cause any problems? Probably not. But you never know. So, with that little thought about drugs, I will leave you. If you like what you heard, saw, then please subscribe, and I'll see you next week when we look at some more GCSE biology, topic by topic. Take care, stay safe, bye bye.